Hi, I'm Yidan Zhang. I'm a first-year PhD student in the Department of Earth and Environmental Sciences. Today, I will show our study on associations between halogenetic flame retardants exposure and childhood gut microbiome in the Canadian cohort. So I'll start on a little bit background of this study. An imbalance in the childhood gut microbiota has been linked to many health risks like metabolic disorders. And halogenetic flame retardants are a group of chemicals that used as additives in many products like cars and furnitures for fire resistance. Previous studies show that early life exposure to HFRs may alter gut microbiota in animals, but those studies remains underexplored in population studies. So this study aims to examine the associations of HFRs in children with their gut microbiome in first cohort in Canada. So here's is our workflow. And our population is 253 children with 8 to 12 years old and with homogeneous population characteristics. For HFR's detection, we use two biometrics here, plasma and stool. And in plasma samples, we have detected 5 compounds of HFRs, and in stool samples, we detected nine compounds of HFRs. And for the gut microbiome side, we also collected the stool samples and did metagenomics and metatranscriptomics sequencing to get the relatively abundance of species and functional pathways in gut microbiota in children's gut. And after gathering those data together, we used linear regression to screen the associations between concentrations of HFRs and also relatively abundance of microbiome features, including species and functional pathways. Models were adjusted as for several covariants for controlled confounding factors. And in the study population panel, we have shown some information of our population and also exposure levels of HFRs in both plasma and stool. And moving to our results part, in this forest plot, we can see the blue bar shows that in stool samples, BD99 and TDP were associated with were negatively associated with those two species that's related to carbohydrate metabolism and also short-chain fatty acid producing in gut. And also in plasma samples, BD209 in this orange bar you can see was negatively associated with these species that also related to short-chain fatty acid producing function in gut. And moving to this panel, those two volcano plots shows the associations between HFRs and microbiome functional pathways. In the first panel, you can see stool BDE 153 was positively associated with those three functional pathways that represented by red dots here. And those functional pathways are related to GABA degradation and gum barrier functions and also energy metabolisms in gut. And also BDE 153 was negatively associated with with this one functional pathways that related to lipid absorptions and obesity in children. And also in the second panel, stool TDP was positively associated with those two functional pathways that related to GABA degradation and gum barrier functions in gut. And we did not find any significant associations between plasma samples, HFRs, and functional pathways. So basically, in this study, we can see that in children from this Canadian cohort, both plasma and stool HFRs exposure was associated with alterations of gut microbiome profiles, with stronger associations observed for stool HFRs. And the downstream effects of those alterations are way to explore. Okay, that's my poster, and thank you for your attention.